Thank you everyone for coming today, and keyboard. And as you all know, work of this nature is always built on the shoulders of giants. So I'd like to start by particularly thanking our labs. I'm in Steve Peterson's lab at WashU, and Matt and Tim are both in David Pennison's lab, also at WashU. We'd also like to thank Mark Rabel's group and the fMRI at Oxford for technical help at various points in the past as well as in the recent months. So as you've heard recently, there are multiple ways to define functional areas. Unfortunately, these aren't all available in humans. And today I'm going to talk about our efforts using both resting state functional connectivity MRI as well as diffusion tractography MRI. Now, the first one of these methods, and I realize that a lot of the methodology we've been hearing about today is DTI. My personal expertise is actually functional connectivity. But this is based on the observation that time series across the brain actually have some structure in their correlations, and it seems to reproduce functional relationships, functional networks, and organization in a spatially defined pattern. So you can actually take these correlations and display them spatially. So here I'm showing the correlation between a particular seed's time point here in the angular gyrus and the time courses from all the other boxes in the brain. So for instance, here you can see it's negatively correlated with this area here in anterior insula. Now, the reason we think that this method will be one of the ways that will probably help define functional areas is because if you place two seeds that are spatially quite close to each other, the maps can look quite different. Here you can see they're actually quite different. You can see they're contralateral homologs from each seed as well. Now, the other method that we're going to be talking about, of course, is diffusion tensor imaging or diffusion tractography. And this is based on the observation that, as we heard earlier, water likes to diffuse more along axons than across them. Now, for the competition, we used the HARDI dataset, the diffusion-weighted imaging with high angular resolution. And we also had to apply a sort of ad hoc distortion correction. So as you can see here, looking at the corpus callosum, the alignment pre and post distortion correction, we feel that this very much helped our results on using the DTI. Now, for the functional connectivity, I should point out. I'm sorry. I should point out that we only had 5 minutes of data provided for the contest, but in my own work, we're actually using a fair bit more, although once you get above 10 minutes, it's probably pretty good. So for the DTI, to generate maps, just as we do with functional connectivity, you could place a seed on the white matter, and I'll get into how we generate these services in a moment. And then from each seed voxel, we initiate probabilistic tractography along multiple fiber directions, and then combine the results using just a weighted sum. Now, you can see here we have a seed in Wernicke's area, and it illuminates the arcuate leading up to Broca's area in the front. So just as with functional connectivity, you can put two seeds that are quite near each other here, approximately similar to the areas I showed you before, and you can see that there are very much differences in their connectivity maps, where you saw before in the correlation maps. So we have two separate methods that can give you spatial differences in their whole brain pattern, or the surface field pattern in this case, based on the exact position of their seed points. But the question becomes, what happens when you move from this seed point to this seed point? And so you can calculate the difference between these spatial maps using an eta-squared metric, which calculates exact difference or similarity between two time series, or you could even just use a spatial correlation. Now, for sorry, I'm stuttering today. I'm going to be using functional connectivity for the next portion of the talk, but the same logic absolutely applies to both the functional connectivity data as well as the DTI data. So to ask, do functional connectivity maps or DTI maps change abruptly between functional areas, you can just simply plot a line between these two regions and directly ask this question. Now, this is important because if the functional connectivity maps or DTI maps change in an abrupt fashion, you can use this just as tracer experiments were originally used to find the borders between functional areas. But if it changes just gradually, like a gradual drop-off of distance, you're not going to be able to independently detect the gradient position between them. So for each point along this line, you can generate seed points and then generate functional connectivity maps for each one of those points. Then using an eta-squared statistic or spatial correlation, you can compare how similar these maps are to each other. So for instance, here we have a slice from the angular gyrus correlation map, and it's very similar to the seeds that were placed along the line right next to it. And then you get this rapid drop-off and then over to where the supramarginal gyrus is. Now, one small note, eta-squared ranges from 0 to 1, with 1 being identical maps, 0 being as dissimilar as possible, and 0.5 being statistically independent. So you can see already that the angular gyrus and supramarginal gyrus are statistically independent from each other. And the same happens in reverse as well. So we seem to have a small region in between the angular gyrus and supermodern gyrus where the maps change pattern rapidly over the entire volumes that are being compared. So if you wanted to just draw lines between every two possible putative functional areas that are known, this would take forever. So instead, you can just examine the entire cortical surface at once. So we generated white matter and gray mid-thickness surfaces using FreeSurfer for each of the subjects in the competition. And then we also registered each of our subjects to the PALS Surface Atlas using Caret, because this allows us to use a common set of surfaces, a common set of grids for each and every subject, as well as gives us from FreeSurfer as well, it gives us a one-to-one concordance between the white matter surface and the gray matter surface. This also allows us to unfold the cortex into a flat plane, because obviously dealing in 2 dimensions is much more computationally facile than dealing with a 3D surface. So, just as we did before laying down a line of seeds, now you generate a whole surface of seeds over the entire left hemisphere in this case. And just to orient you, if you haven't looked at these too much, this here is the sylvian fissure, central sulcus right in the middle, that makes this the frontal cortex, parietal, occipital lobe here, and temporal lobe over to the side. So I'm showing you here the correlation map from this particular seed, which is shown right here in the flat map, just as I showed you before. And now we're going to compare it to all other correlation maps generated from all of the seeds across the surface. And we're going to plot the values over here on the right. So we're going to take this location here, generate its correlation map, compare these two correlation maps with eta squared and get a 0.5 value. These are the same maps I showed you before. And then plot that value back here on the right as a color. You can then do this for all of the maps compared against this particular correlation map. And now you've generated an eta profile for this particular seed. So you can see that there are stark transition zones, as well as there are areas far away that have very similar patterns as well. For instance, this is the angular gyrus region and the posterior cingulate, both involved in the default network. So now to find the locations of rapid change here, you can simply take the first derivative of this map, the gradient image, for instance, and you can see that you can identify the sharp boundaries. Now, unfortunately, this process generates thousands of eta maps and thousands of gradient maps because we have thousands of seeds. So here you can see just a sampling of them, and you can see that a lot of them have quite different patterns. If you average all of these together, you can get a putative border map or probabilistic border map for functional areas across all of these maps. And so when you cross one of these lines, the functional connectivity maps change quite rapidly, or not so much, depending on the strength of the line. And of course, you can project these back into the brain. Now, the work I've shown you so far was actually my own thesis work that I've been working on prior to the contest. So if we turn to the contest data and incorporate some improvements I've made in the method since then, you can see that on the left, for brain 0, we have the consistent boundaries generated from functional connectivity maps, and on the right you have the consistent boundaries generated from DTI maps. Now, these do look quite messy, but there is a lot of data in here. So again, what I just showed you was the borders between functional areas, but we're not actually interested in the borders per se. We want the functional areas. And so if you think of not edges as holes, and there's areas there, you can actually directly examine those. So for instance, we'll take the average gradients from one subject, flip the image over, so now hot equals consistent functional connectivity maps, just as you'd expect if you put two seeds in the same functional area. Blur it slightly to find the stable peak within those holes. Find all those peaks using just standard, local extrema finders from MATLAB. And then you can then flood fill from each one of those positions with respect to the local gradients around it, and you generate functional areas on this flattened surface. Of course, these can be projected back onto the surface, so I'm showing you for one particular spot here, as well as projected to the volume, because we've registered all these surfaces to the volumes as well. And these putative ROIs, which I'm showing you here for brain 0, also seem to align with the scoring ROIs on the surface, as well as my own functional localized data I have on other subjects. So for instance, here's like the tongue representation of left motor, and I apologize, red is now good and green is now bad, because of the color maps we have available. And so you can see that we do seem to find one that's right there, right in the middle. Left Broca's area, when you intersect the functional localizer data with a cortical surface, you do separate this into 3 spots, probably because the hot spot is right in the center, given the lower resolution of the fMRI. And you can see we actually identify those 3 peaks on the surface as well. Also for V1, you can see that even though this is getting split, these are all just settings that we need to change in this. We've done in a rapid fashion, of course, for the contest. We see we've identified some areas here which are probably V1, and if you look just above that, you can see that this next strip is probably